Welcome everybody to Cold Spring Harbor Laboratories Live at the Lab. I'm Aline Augenbraun, Creative Director for Public Affairs at the Lab, and today we're talking to Amanda McBrien about the innovative ways she and her colleagues at the DNA Learning Center kept teaching hands-on science during the pandemic. The folks at the DNA Learning Center have been innovators in science education for about 30 years, and I think it would take more than a global pandemic to stop them from innovating now. We're going to be taking questions during the webinar, so if you have any, please click on the Q&A button below, and we will get to them soon. Now, let me introduce you to Amanda. Amanda McBrien is Assistant Director of the Cold Spring Harbor Laboratories DNA Learning Center. She leads the educational programs. She's translated her lifelong love of animals and biology into teaching students and their teachers about genetics, environmental biology, evolution, and, many other things. Her approach, like that of all the educators at the DNA Learning Center, is innovative and hands-on. This month, she was named in Crain's New York's Notable Nonprofits and Philanthropy list. Congratulations, and hello, Amanda.
Hello, thank you so much. It's a pleasure to be here.
So tell me, for people who don't know, what is the DNA Learning Center and why is it important to teach the way you do?
Well, what I'm going to do to answer that question is I'm going to share my screen and show you a few slides that illustrate what we're all about and the types of programs that we've been offering here in New York for the last 30 years. So let me share, and we'll jump right to it. So for those of you who may be new to Cold Spring Harbor Lab or to the DNA Learning Center in general, the Learning Center is an operating unit of Cold Spring Harbor Laboratory, and we specialize in education and outreach. Our goal really is to educate, inspire, and engage with teachers, students, and the general public through hands-on laboratory experiences. Here in New York, we're operating currently out of 4 different locations. On your screen right now, you see a picture of our location in Cold Spring Harbor, the Dolan DNA Learning Center, which is our flagship. We have 4 teaching labs, a museum exhibit, a bioinformatics or computer lab, teacher offices. But we have other locations that you may or may not be familiar with here in New York. The DNA Learning Center West is a location that opened in 2001 in collaboration with Northwell Health in Lake Success, which is right on the border of Nassau County and Queens. Unfortunately, it closed in 2020, nothing to do with the pandemic, but the lease of the building that Northwell was using to house their clinical lab expired. So we had to clear out in May of 2020, tough times, and we're now awaiting to find out whether or not we'll be able to relocate to a different Northwell building. But that location served us well because it was kind of a gateway to Long Island from the boroughs. So we were able to start hosting field trips with high school students and middle school students from New York City at DNA Learning Center West. After West opened, we spread to the city and opened the Harlem DNA Lab in 2008. And if you look at the building, you'll see it's actually a school building. We have a renovated lab space in that school building. It's actually an educational complex where there are several schools housed. We received that space from the Department of Education as part of a grant to train teachers in New York City to implement genetics and biotechnology labs, and now we use it as a field trip location as well. Next came the Regeneron DNA Learning Center, which is indeed a licensed center, licensed by Regeneron Pharmaceuticals, and our center is on the Sleepy Hollow campus up in Westchester County, so it allowed us to reach districts outside of the boroughs and further up into northern New York and western Connecticut. It did open in the fall of 2019 and was operational for a few months before the closures, and will be reopening in fall of 2021. We are very much looking forward to that. Finally, the DNA Learning Center NYC is our newest location in Brooklyn, and it is going to be a standalone center that I'm going to tell you more about at the end of the presentation. But you'll see a picture of a building here. This is a building that belongs to City Tech, which is one of the campuses of the City University of New York, and we'll be occupying the whole second floor of that building by the summer. We have been using, though, a temporary lab space on the 4th floor of that building, and we've been hosting field trips there since early 2019 as well. So on to programs. The DNA Learning Center started offering teacher training programs back in 1985. Our director, Dave Miklos, began by training local teachers and teachers across the country in basic biotechnology techniques that he really felt belonged in the classroom. Since then, we've trained over 15,000 teachers in different techniques, usually through grant-funded programs. Our most recent initiative on Long Island has been in DNA barcoding. But we don't just train teachers, we also develop resources for teachers. For decades, we've developed kits that are distributed through one of our longtime partners, Carolina Biological. And we've also developed a collection of websites, apps, textbooks, and even sites where streamlined DNA, high-level DNA analysis can take place. What we're probably most well known for here locally are our in-person student programs. So we work usually with about 30,000 students a year. About 2/3 of them are visiting one of our centers for a half or a full day lab field trip. These are booked by teachers and school administrators, and the content is chosen by the teacher. Generally, labs are selected that align with curricula in middle school life sciences, living environment, which is high school biology here in New York, advanced placement biology, or other advanced electives. About 1/3 of that 30,000 are seen in school, so we can also pack equipment up and bring the field trip to the students. On top of these in-person programs, for a long time we've had week-long summer camps at our centers here in New York. These range from introductory courses like Fun with DNA, where students learn foundational concepts in cell biology and genetics, all the way up through very advanced courses in genome science and DNA barcoding. The most recent addition to our local programs are our student research initiatives. They're all in barcoding. The Urban Barcode Project was our first, which started in 2010 in New York City, followed by the Urban Barcode Research Program, which is also in New York City, and Barcode Long Island. Two of these programs have a teacher training model where we train the teachers to become mentors for students who are conducting authentic research in DNA barcoding. We not only train them, we provide access to equipment, reagents, kits that can be borrowed and returned, open labs, access to our faculty for troubleshooting, help, and advice, and a symposium at the end of the year's work. The Urban Barcode Research Program is a slightly different model. Instead of training the teachers, we train the students. They get 2 weeks of training in DNA barcoding and conservation genetics, and then they get paired with a partner and a scientist to conduct their research during the school year. So, all told, if we were to look at an average year, pre-pandemic, and count up the lab exposures, including students who are visiting us for in-person programs or students who are accessing programs at other DNA learning centers that are licensed to us or have been modeled after us, there are 20 of them around the world, or students who are using resources we've developed. In an average year, we can estimate that almost 570,000 students a year are doing hands-on lab experiences or online bioinformatic analysis using our stuff. So I'm happy to report to you that we are very proud of what we do, and you're going to find out soon how we tried to keep this up during a very hard year.
It was a very hard year. So, I mean, all this hands-on stuff. Before we go forward, can you just explain a little bit about the barcode project? Because that's an amazing citizen science project, but I'm not sure people know what a DNA barcode is.
Sure, I do have slides about that I can show later, but I'll give you an overview now. So, DNA barcoding is a technique where pretty much anyone, a citizen scientist or a scientist, can extract DNA from a tissue sample, whether it be from an animal or a plant, any living thing. And a technique called PCR, which has become very familiar to everyone in the world in the days of COVID, to amplify or make lots of copies of a specific region called a barcode region. And that barcode region is a DNA sequence that's unique to that species, just like a barcode on a package at a grocery store. So you scan it, the machine knows exactly what it is. Well, with a DNA barcode, you get a DNA sequence that's a few hundred bases long, and you can do something like a Google search with it online, and you can see if there are any any matches in a database that's been generated by scientists. And with this information, you can figure out what that living thing is, sometimes all the way down to the species level, or at least what it's related to. So it's a technique that's often used in conservation biology, conservation genetics, but also in fun fields like food fraud.
Yeah, I mean, your students actually found some interesting stuff about fraud in our urban food landscape. You want to tell one of those stories?
So in the early years of the Urban Barcode project, we had a few interesting food fraud projects. I think one of my favorite had to do with bird's nest soup, if you're familiar with it. So there were a few students at a school in New York City, Trinity School, which is an independent school, who were both Chinese and were from families where bird's nest soup was a common remedy, almost like a wives' tale type item. In fact, their parents believed it to not be true, but that these bird's nest soups were actually made from bird's nests, from this little bird called a swiftlet. They actually use their saliva to build these nests. So a lot of apothecaries sell bird's nests, different types of bird's nests that people spend a lot of money on to make these homemade remedies. These kids went out and bought a bunch of bird's nests from these apothecaries and extracted DNA from the bird's nest and actually proved that they were indeed from swiftlet nests. I mean, this has nothing to do with whether or not they have medicinal properties, but both of these boys reported that their parents were sure that it was a lie, that these apothecaries were selling fake bird's nests, but it actually turned out that they were legit. They were swiftlet nests. So a cute little story with an interesting ending.
So all this interesting hands-on stuff, did it stop during the pandemic?
It did not. There was a temporary pause, but then we picked right up when summer began. So if I may, I'll pull up some more slides and start telling everyone about just that. All right, we're back to slides. So in March, we were faced with the same thing everyone here in New York was faced with, sudden closures. And being a hands-on science center, this was especially troubling because how do you operate a hands-on science center without the hands? No students were going to be able to come to us for some indefinite period of time, and we certainly weren't going to be able to go to their schools. And at the same time, all of the teachers that we love to work with and support as best we can were now being thrust into this new educational landscape of teaching completely remote. Some had an easier transition than others. We were very fortunate that we kind of had a leg up when this all happened here in New York, because in February, prior to the closures here, we did a few live stream events, some lab demonstrations for schools in China. So one of our licensed centers, DNALC Asia, reached out to schools they knew, and we offered over February break, so just weeks before our own closure, 2 lab demonstrations, a bacterial transformation and a restriction analysis, 2 very popular biotechnology labs. Each were an hour long, they were done on Zoom, and total 1,400 students from China joined us for these 2 sessions. And it was all the rage. They were very active in the chat, super excited to have some kind of engagement because they were stuck home doing lots of boring work, it sounded like. So we were able to do this pretty quickly because we have some very talented staff people here who had already done a lot of multimedia production for us. In fact, I'm in a studio right now. We have very nice cameras and equipment to be able to provide something like a live stream lab demonstration. So because that was so successful and it went pretty smoothly, we knew in March when the schools here closed that our first step for the schools and the students here on New York was going to be to provide something similar to that, but on a larger, more regular scale. And thus was the birthing of DNALC Live, a site where every week we posted 12 to 15 live sessions. Some were short, some were long, many of them were filmed at home. One of the first things we did when we knew the DNA Learning Center was going to close was instructors scavenged the building, grabbed as much lab equipment as they could, reagents to do experiments, webcams, laptops, so that we could settle in at home and start providing some resources for the teachers who needed them the most. In addition, we saw this as a real chance to expand our online footprint a bit, something that we hadn't really devoted a lot of time to in the last few years. So I have a slide that shows some of the examples of these sessions. So these were all filmed on Zoom but streamed live to YouTube, meaning anyone who was on YouTube at that time could go into a chat and engage with an instructor. So generally there was an instructor teaching and a second instructor in the chat answering questions and responding to comments. It worked really well, and it was good practice for what was next to come. But we had some sessions that were short, so little animations of things like, why does soap work when you're trying to get the virus off your hands? Interviews with scientists or other people with interesting STEM professions. Tours of our exhibit, which have become very popular still on our YouTube channel. The lab demonstrations that we actually started with China. In fact, we pretty much recorded our whole catalog of field trip labs, and they've been archived. And then there were some fun series about things like bugs. Now, who would want to do that? One of our instructors has a background in entomology, so he was able to produce a really great segment about all of the bugs in our backyards. Our evaluator has a background in anthropology, so she did a series called Species Shorts. The AP Biology test was coming up, so we set up some live chats so that AP Bio kids could connect with each other and connect with one of our instructors who was a former AP Biology teacher. So all told, by the end of May, we had recorded and archived over 100 of these sessions. And we did indeed increase our footprint. So I just have a few graphs, I'll go through them quickly, that show our YouTube views increasing. So the X shows when the pandemic began. So pre-pandemic, views per month, post-pandemic views per month. So there are some ebbs and flows depending on seasonality. So kids were still in school at this point, then it was summer. And then they went back to school. And now we're seeing an extremely large peak because our museum tour is somewhat of a blockbuster, hundreds of thousands of views. In fact, we think YouTube might be helping us a little bit by recommending that video to other people watching videos similar. Our subscriptions to our YouTube channel also increased substantially, and you can see pre-pandemic and then post-pandemic. In addition, view times increased. I think the average view time right now on our YouTube channel is 10 minutes, which is much longer than average view times. And our engagement with the public through our YouTube channel has also increased. So the number of comments when we were doing DNALC Live substantially increased from the tens to the thousands. So we certainly met our goals and also provided a lot of great resources that we can see teachers are still using this year. So that was step one. Step two was starting to teach real students. So we got really good in April and May at teaching invisible students on YouTube that we couldn't see. And it was a real thrill to start preparing for a summer camp season. But again, how do you do this? How do you have these immersive, experiential summer camps without kids in your building? Well, luckily, we had experience developing kits, and part of developing kits is developing robust protocols that we know will be successful in the hands of middle school and high school students. So we came up with the idea of having kit-based summer camp season. We had already set up our labs for our live streaming, so the cameras were set up, the computers were set up, we had that infrastructure already from DNALC Live. The only thing we added was a big TV screen in every room so that when we were teaching we could see our students and interact with them and engage with them. We also decided in addition to doing live camps where students would zoom in and participate with their classmates, we would offer an on-demand option. That would be pre-recorded video along with a materials kit or a kit of materials that could be done self-paced. And it would include also access to a Google Classroom so that participants could still communicate with us as needed. But more than just this classroom infrastructure, we spent a lot of time developing kits. Our whole building became Kit Central. So I took a picture of this sign because it just made me laugh when I saw it in the teacher's office. But we have become experts in packing kits. We've packed thousands of kits. Over the summer, it was more about 460 kits, but 460 students taking our camps was a really pleasant surprise. It's about 50% of what our typical enrollment is in summer camps, so we were very pleased. These are all middle school kits, so we adapted the camps that we could and came up with materials lists to support students doing all the labs that they possibly could as part of the camp curriculum from home. For our middle school camps like Fun with DNA and Forensic Detectives, it was a little bit easier than it was for some of our more advanced camps like genome science and DNA barcoding. They're PCR based and have a lot of bioinformatics, so they were actually a good fit for this transition to remote. But one important step that had to be completed before we could do those high-level camps was coming up with some new robust protocols for DNA isolation. So we had a team of instructors who, instead of focusing on DNALC Live in the spring, focused on development and testing of protocols. In a very short period of time, they managed to come up with several DNA isolation protocols that were used for kids to extract DNA from their own cells, from things like Doritos to check to see if they're genetically modified, from invertebrates and plants for DNA barcoding. And then we came up with a system in all of the high school camps of spaced instruction so that post-DNA isolation at home, samples could be mailed to us very quickly, overnight, and the following day of instruction would include the processing of the samples. Normally, the processing, which includes steps like PCR and gel electrophoresis, would be done by the students. Those things had to be done on site by our instructors, but they were done in real time with the real samples, so kids could actually watch along and see their samples going through this set of steps. And the end result is a DNA fingerprint, a visualization of the DNA that is data that can be used for further investigations on things like population genetics, or a DNA sequence that can be used for DNA barcoding or to answer questions about human origins. So this was really an organization-wide effort. Our biomedia team was setting up infrastructure for us. Our administrative team was handling registrations and a million questions from parents. Our educators were adapting these courses, and then our development team was reimagining the labs so that we knew that we'd have success. It was an amazing summer.
It sounds amazing. Amanda, we have a question.
Sure.
Were the Doritos genetically modified?
So I believe so. So it's the corn, and yes. So I think they usually are. There's an organic type of Dorito. I don't know if we've tested that one, but you can even look on the back, and I think on the label it even says that it's made with genetically modified ingredients. Good question.
Yep, I think you've engaged your audience.
With Doritos, it's so easy. So after a successful summer, we took everything we learned, all of the new protocols, our kit packing skills, our engagement with students in remote classrooms, which was also a really steep learning curve for especially people like me who are technologically disabled. It was hard, and I feel like by the end of the summer, we were all feeling really great about remote instruction. And then the school year came, and it felt very different in the fall, and I'm sure if there are teachers here, they would agree. In the spring, it was just shocking. Everyone was home, but everyone was in the same boat, right? Everyone was home, completely remote, no ifs, ands, or buts. But in New York, when school opened in the fall, it was, oh, some kids are coming back, some aren't coming back. When they come back, they're going to be in small groups, so they'll be in a day or every other day or 2 days and then out for 3. Every district had its own, variation on this hybrid blended learning theme, which really made things difficult for teachers in school. Things were pretty quiet for us in the fall. We were developing because what we really wanted to do was be able to provide these teachers with some field trip options. We spent time talking to some teachers about what they wanted, what they needed in the fall. It was clear that many of them were very overwhelmed. Many of them were not doing hands-on labs. They planned on doing them eventually, but we're still just trying to navigate the landscape. So in the fall, the 3 field trip options that we came up with were live field trips, just like our live camps, where a teacher could get a package of individually packed kits, distribute them to students, and then on Zoom, they could do the wet lab portion, the DNA isolation, let's say. The teacher could collect the samples, send them back to us, and then we set up future sessions for the processing and analysis steps. We also did on-demand versions of all of our lab field trips. So again, pre-recorded instruction of that single lab accompanied with kits so the samples could be collected and returned to us for processing. We thought that would be a little bit easier for teachers who were having difficulty scheduling with irregular class schedules. And then live demonstrations, which actually have turned out to be the most popular option. They're shorter, they don't use the kits, but they demonstrate all of the lab techniques. So these demonstrations are a lot like what we did on DNALC Live, but the difference is you're with a live instructor and you're watching things in real time, and you can engage with that instructor and ask questions and answer questions like you would in an in-person classroom.
Amanda, you told me a story once about how you were engaging the kids to watch their own samples being processed, that was more engaging. And we have a question from the audience, actually, about engagement. So they're asking, how was the engagement level of the students with the virtual format? I mean, people assume they'll be less engaged. But what have you found? This is a very interesting test case for many people's assumptions.
Yeah, it's a great question. And it's something that in the spring when we were preparing for our summer camps that we were extremely concerned with. A lot of us have children, and we were watching our children at home in remote classrooms, not being engaged all the time. It's hard. It's very hard in a virtual classroom to maintain engagement. And it's also hard sometimes to even know if you are, especially if cameras are off. So there's this whole issue where cameras on or off. During our camps, we encouraged every time we could for kids to have their cameras on unless they felt uncomfortable. It was much easier to get the younger students to comply with that, especially when they were in breakout rooms, because that's when they could really talk to each other. But we've made every effort, especially with our camps, because we had them for extended periods of time, and we were really trying to build a sense of camaraderie among the classes like they have during a regular camp, to include techniques like breakout rooms, for example, and come up with creative ways that kids could work in teams, just like they would at a lab bench in teams, completing a task or an activity and then reporting back to the class. We started using some apps that are used generally for informal assessment, but they're fun ways in an anonymous way to get kids to interact with you so you know that they're paying attention and that you know that they're getting what you're teaching. For me, that was the hardest thing. Are they really understanding what I'm teaching? It looks like they're paying attention. Their cameras are on, but sometimes I would do one of these fun, there are these things called Kahoots, a little assessment at the end of the day, and maybe 50% of the answers would be incorrect, and I would make sure to go back the next day and review those things. But yeah, engagement is hard. I think the nice thing when you're using kits, though, is that they're doing the lab along with the instructor, and that in and of itself is engaging. Or if they're doing analysis, same thing. They're doing it themselves along with the instructor. They're not just listening to us talk.
Yeah, that's what one of we have another sort of comment here is that, if the kids are doing it themselves, like they have to do it at home in order to get to the end, like they can't rely on somebody else. They have to do it themselves. And that may actually be an interesting technique for the future.
It could be. I have to tell you, I'm a middle school teacher, so I'm used to reiterating things 10 times. I'm used to moving around a classroom and making sure everyone's on task and answering the same question, a million times. Can I eat it? No. But when you're in a virtual classroom, it's a little bit different. You can't hover the way that you normally would and make sure that everyone's on task all the time. So they are, you're right, a lot more independent in many ways in these remote classrooms. So on top of the field trips, there are a few other interesting things, and this comes back to citizen science and research, that we did continue during the pandemic. So in addition to coming up with these new virtual camps and the kits and the protocols to support them and then the field trips to follow, we did continue with our student research program. So all of our barcoding programs started normally in 2019 and then finished abnormally in June of 2020. About half of the participants who started projects in September, were able to complete them and present at a poster session in June, which we were really thrilled about. We did a virtual poster session. It was set up on Slack. Every team that had a poster was able to present the poster, and visitors, including staff at the Learning Center, which was really fun, were able to go from room to room and actually engage with the kids who did the projects, just like we would in a regular old symposium. And then in September, we started a whole new cohort of students, starting a whole new set of projects that will be completed this June, and we're going to be doing another virtual symposium again. It was quite successful. So we continued to support our student researchers, and then we continued to support citizen scientists. So there was one program called US Ants that actually started and finished during the pandemic. And this is not a program that I oversaw, but it's just an amazing feat. During the summer, we had a small pocket of money to reach out to Audubon societies, Sierra clubs, nature sites, you name it, we reached out to them to try to find people who would be interested in collecting ant samples and helping us contribute to the understanding of the over 900 species of ants there are in the United States, and to the data sets of barcodes that we have for these ants. And over that short period of time, in less than a year, I have notes because sometimes I can't remember the numbers. 322 ant samples were collected and processed by citizen scientists who were using kits that implemented some of the robust isolation protocols that we developed for our other programs. Some of these participants even received equipment kits that they borrowed and returned to us. And they did some of the processing of the samples themselves, the electrophoresis. Many of those samples, if they look good, got sent back to us and eventually were sequenced. And interestingly enough, there were 56 species of ants identified from these citizen scientists. 18 of the sequences that were generated are potentially novel sequences, meaning they don't exist in the databases yet. So they'll be published, if they turn out to be novel sequences, by these citizen scientists as authors. And there were 10 species that were undescribed, meaning we're not sure if these are ants that people were even aware existed. So we have experts vetting those samples. But these are all people, non-scientists, who have now contributed to our understanding of ants and their importance in ecosystems and as indicators of habitat destruction, climate change.
Amanda, I'm sorry. I have a question here. Do the discoverers get to name the ants, the new species?
You know, that's a good question. I'm not sure if we've had a case where that's come up, but that's very common in science, is it not? If I were the person who found the ant, I would certainly want my name incorporated. That's my vote. But you can certainly get your name published on the barcode that goes into GenBank, into the repository where all the sequences live, which I think is also super cool. The other interesting thing that we started doing in the fall, and this was actually supported by funding from the National Science Foundation through a collaboration we have with Innovate Bio, which is the National Biotechnology Education Center, is kits provided to college faculty, specifically community colleges, some high school teachers. We reached out to college faculty we knew well because college teachers teaching college courses in biology not being able to get into their labs is a travesty. We know that understanding and learning science is done best by doing science. So we were hearing tales of community college professors not being allowed to even go into their labs alone to retrieve samples or to collect some equipment or materials. So we reached out to people we knew, and over 800 students are now using our barcoding kits and kits to extract their own mitochondrial DNA that were developed during the pandemic and were sent to these faculty to distribute to their students so that they could have some hands-on experience and some fun analysis of data that they generated themselves.
We have another question about, this does bring up an interesting question. The particular question is, was US Ants funded by the NSF? And it also, like these citizen science projects, without the citizen scientists, like a lot of environmental projects can't happen. There's not enough money on the planet. But tell us about US Ants.
So US Ants was actually, I consider it an offshoot of Barcode Long Island, which was funded by NIH. So US Ants is an NIH program. NIH has these subunits, and I'm not remembering the acronym, which is very bad of me. But it's under the umbrella of NIH, just like Barcode Long Island was. And it was a small project that actually, believe it or not, was originally funded for something entirely different. There was going to be an exhibition in Washington, D.C., and we were invited to participate in this exhibition and bring some students who were part of our Barcode Long Island program to set up a table. They were going to have a Lego sequencer. This was all in the works. It was very exciting. And then COVID. So we were asked by NIH to come up with a different plan, another way to spend this small pot of money. And having a campaign like this made a lot of sense. It was something that was very doable in a time when traveling was difficult. So we did this really great hybrid model where all of the training was done virtually. And I'm sure this is something that will carry forward for some of our projects. You know, 95% of these citizen scientists weren't from New York. They were from out of state, and we trained them, and they were really successful.
I got to ask you to back up. So, Lego sequencer?
Yeah. Sharon Pepinella, who manages our research programs, was connected with a group in Europe that had come up with a template to build a DNA sequencer made of Legos. She actually built it. We had purchased the Legos already. We have it, and you can put Legos in, and then you get a little Lego sequence out. That's all that I can tell you because I am not an expert in Lego sequencers, but it was going to be part of a table at an exhibition where kids could come and learn about DNA barcoding and even do a DNA isolation and a simulation of sequencing in this Lego sequencer.
That sounds amazing. How can your school get involved? I mean, how does a school get to do any of this stuff?
So our local programs are easily booked. If you just go to our website, there's information about camps, there's information about field trips, in-school instruction. All of our programs are for grades 5 and above. So all the information you could ever want about the programs themselves, the labs we offer, and registration information, which is generally done by phone, is on the website. Look, this is the sequencer. Someone brought it to me.
Oh my gosh, that's amazing.
In case you didn't know, it exists.
You know, you also had another story that I thought was so fascinating. You know, like we think of lab equipment as being so fancy and you know, expensive but you were talking about doing PCR at home.
I was. Yeah, so I don't have a slide for that. You caught me. So one of the development projects this spring was to use a technique called Isothermal PCR. I was not the developer of this technique, and I did not implement it, so I'm not going to be able to give you super rich details. But PCR, polymerase chain reaction, is when you take a piece of DNA and you use an enzyme called Taq polymerase. It's the enzyme that copies DNA in cells. And you can target or bracket a region of DNA and get the enzyme to make lots and lots of copies of just that region you want to look at, that needle in a haystack, right? So like you're a geneticist, you want to look at one region of a gene where there might be a mutation, not all of the genes. So you can use PCR to find that region and make lots and lots of copies of it. To do that, you put everything in a tube, and then you put the tube in a machine called a thermal cycler. And what the thermal cycler does is it cycles through a series of temperatures that get the DNA to unravel and split apart. It's called unzipping, so that the enzyme then at another temperature can bind to the DNA and start to make lots of copies of it. So you go through a series of temperature cycles over and over and over again to keep repeating this process of splitting the DNA, binding these little pieces of DNA called primers that tell the enzyme where to go, and then getting the enzyme to copy. So thermal cyclers, you know, the ones that we use, cost about $5,000. I think in the early days of PCR, scientists used water baths that were set up at these temperatures, and you'd have to go a minute and a half, a minute and a half, and a minute and a half, and that's cycle 1. a minute and a half, and you do cycles, maybe 30 cycles, to get lots and lots of copies. Isothermal PCR eliminated all of the heating and cooling and set up a reaction in the tube where instead of cycling through temperatures and using the enzyme to make lots and lots of copies, it usually takes about an hour and a half, and then using a technique called electrophoresis to see the results. It allowed students to be able to set the reactions up in a single tube and wait for a color change that indicated the presence or absence of that snippet of DNA that you were looking to identify. So it's a technique that's already been developed, and what we tried to do in a very short period of time is develop it to be robust enough for students to do. And we had some success in our summer camps with kids doing an isothermal PCR reaction in genome science. It turned out to be a little bit more finicky than we thought, but a really fun, brand new technique for the DNA Learning Center that became part of one of our camps.
That is amazing, and that's there's another question that's kind of related to that, which is, besides the citizen science stuff, a lot of these virtual formats and the kits you've developed, are these going to now become permanent? Is virtual going to be part of the new world order?
I think so. I don't think it's going to go away. Certainly this summer, we're advertising in-person camps. At Cold Spring Harbor and Brooklyn, we'll be following CDC regulations, so probably our capacity will be somewhat limited. But we are also offering all of our virtual camps, multiple sessions, and people are signing up for them, either for geographic reasons, because they're from out of state, or for personal reasons. So I don't see why that wouldn't continue. I also don't see why we wouldn't continue doing virtual field trips. We didn't have a lot of out-of-state participants for field trips, but I would imagine if it's something that we keep on the menu and that we advertise a little bit better in years to come, that's something that might become somewhat popular as well. Even for local schools who maybe find it easier to have the field trip brought to them on a screen rather than having us come into the school or them visiting us. And then the protocols for sure. So a lot of the development work that took place revealed procedures, robust procedures that are going to be incorporated probably into future projects pertaining to citizen science or DNA barcoding in general.
You've inspired someone. We have another question here. How do you get your job? I mean, how do you get to be a DNA Learning Center teacher?
I can tell you how I got this job. So I actually started working here back in 1998, a very long, long time ago, which is why I have this gray stripe on my head. And I went through a traditional teacher training process. So some of our educators are people like me who went to school, undergraduate, studied science, and then went to a graduate program in education and decided that instead of working in a traditional classroom, that we would prefer to have a non-traditional teaching experience. So there are some of us that are in that category. When we're hiring, it's always nice to hire someone who has that background in teaching and the right personality. But if you have the right personality and no experience teaching or very little, we can train you to teach. I think that we're pretty good at training up teachers. But I think what's most important here is someone's personality. I think that because we're all the time engaging with young people that we're trying to inspire and excite in the sciences, that the most qualified candidates are always the ones that come in for an interview and just entertain me. That's what we're all about, science and entertainment.
Well, I can imagine. I mean, that's I think people get intimidated by science. And so one of the nice things about the DNA Learning Center is that you've been able to find ways to engage people where they're at middle school, high school, and college.
Yeah, I agree. So, and I think especially I see this in our summer camps. So we don't have prerequisites besides age for our camps, and that's really for social reasons, and just so that we know when someone comes in that they're likely to have learned certain concepts in school prior to starting a camp. But we don't require grades in certain classes or completion of certain classes or anything fancy like that, because really anyone can come to a DNA Learning Center camp or a field trip and have an enjoyable experience. We're science for everyone. We're science for everyone here, and you're right, it can be scary. And I think that one of the things that makes it less scary has to do with the other group of teachers that I didn't mention. So the other half of our teachers are mostly people with PhDs, people who went all the way, who worked at the bench, scientists. Scientists can be a little scary, I think. But there are people who are relatable, who can say, listen, I had this great career in science, I love answering questions, I love performing experiments. You could do this too, with a smile and a great personality.
So I think we're going to start wrapping up here. So you wanted to, I think, you know, the future looks bright, it looks like, for learning about genetics and genomics, whether, you know, people are at home or if they can go to a site or a field trip. So you want to tell us a little bit about, you know, a few of the DNA Learning Centers that are going to be available to people?
Sure. So what I'd like to do, if it's all right with you, is highlight the newest DNA Learning Center, because I'm just super excited about it. So there are our total numbers, just for your reference. So far, a little over 12,000 students who've taken virtual field trips, almost 500 who took camps, over 500 who we've been supporting in research projects, cohorts that ended in June and began in September, citizen scientists, and then college students whose faculty or whose professors got our kits. Super happy with that number. So let's talk about our newest DNA Learning Center. So we are in the final stages of construction. Of the DNA Learning Center at City Tech. So as I mentioned, City Tech is a campus from the City University of New York, and the building that we're going to be occupying is on Tillery Street, which is over here on this little map, and Adams Street. So literally the Brooklyn Bridge is to the right of Tillery Street. When you're coming over the Brooklyn Bridge from Manhattan, this is the first building that you see. We're going to have our own entrance on Tillery, and this is the floor plan for the building. Now this is almost 18,000 square feet. This is twice the size of our center in Cold Spring Harbor. It has 6 teaching labs, 3 that seat 32 students, 3 that seat 24, 2 bioinformatics or computer labs, a nice size lunchroom, which is very important to me. It might seem silly that I'm pointing it out, but you need space to eat. Exhibits, offices, 3 prep labs, including 1 to pack kits, and specially designed for our Foot Locker kits, which are these big Pelican cases that we can put equipment in safely. I have renderings of what the space is going to look like. So this is the design of the classroom. It's a lot like the classrooms we have here. It's got a little bit more of a modern edge. All of the classrooms have coat hooks so students can bring their book bags and their coats in the classroom with them. They'll all be equipped with smart boards like the ones we use here. The benches are designed just like the benches we use in Cold Spring Harbor. What you can't see is that there are shelves to store shared equipment. This is a bioinformatics lab. So this is where students are doing bioinformatic analysis of DNA sequences. And then of course the lunchroom, which has this really jazzy design that's almost like being inside a cell. There are even structures in the ceiling that look a little bit like endoplasmic reticula. So our expectation is that our building in Brooklyn's construction is going to be completed by the end of this month. So our New York City staff are going to start occupying the building, setting up equipment and furniture. And this summer, we're going to start hosting camps there. We have a full roster of 30 camps on the docket, and we're hoping to get kids into every one of them. So selfishly, I'm sharing this final slide with you about where to go to find information about signing up for camps. Please share this information with anyone you know who has kids entering grades 6 and above, and let them know that we're getting back to hands-on science at the DNA Learning Center this year, in addition to virtual camps. And especially if they're able to sign up to go to Brooklyn. It's going to be an amazing space.
Can you speak a little bit about about what people who might be interested in live experiences outside of New York can do? There are international DNA learning centers in other countries.
There are. So we have a licensed center in China, in Suzhou, so all the way around the world. There are also several DNA learning centers or science centers modeled after ours in Europe. I don't have a map to show you or name them all, but I will tell you this. If you go to our website and go to the About section, you can read about many of these centers and their locations. And interestingly enough, 3 of our campers this summer were actually international campers, which was quite a thrill. A couple from Canada and I think one from Mexico. And we've had a school in Switzerland purchase one of our on-demand field trips this year.
Wow, that is amazing. Well, this has been a really great discussion. I think there's one more question here, if you don't mind answering. So you once told me the sun never sets on the DNA Learning Center. What do you think the biggest, when you think about the impact you've had this year on the lives of kids during the pandemic? The question is, what do you, how do you feel your contribution has kept kids' education alive?
I think it's a good question. It's a very good question. In my humble opinion, I would have to say that part of the important message is perseverance. So there was never a time where any of us on staff here said, oh, we're just going to throw in the towel. This is too hard. Students can't come to us. We can't do what we're used to the way we like to do it, so we're not going to do anything. And I think that a great message to send to students is that's just not a very healthy mindset. But on top of that, being able to provide engaging experiences in a year where it's possible students were having far fewer engaging experiences has been quite a thrill. And I can tell you from my own experience teaching some of these remote classes, and I'm teaching the easy audience, so these are the 6th and 7th graders who get excited about lots of things. But being in a classroom with them, even when it's remote, and doing a lab with them when it's likely they probably haven't done any or they've done very few hands-on science labs is a thrill. And I'm hoping that they'll remember these experiences they had during this very difficult year and be inspired in general, or maybe to investigate more into the sciences because they had that really cool lab from the DNA Learning Center.
That is a very inspiring message. Well, this has been such a great pleasure. That's all the time we have for today. Thank you so much, everyone, for participating and asking really good questions. I'm sorry, I think there was one that I think got answered during the course of the discussion, but thank you guys so much for participating so much, and thanks for joining us for today's Live at the Lab webinar. There will be the next Live at the Lab webinar will be on April 28th at 5 p.m. We will be focusing on bringing coding to young women and underserved communities. Your colleague Jason Williams will be talking at that one. So, and if you want any more information, there were a number of people who asked for information. For more information on the DNA Learning Center or anything about Cold Spring Harbor Laboratory, please check out our website at cshl.edu. That's cshl.edu. You can find news, events, everything related to the DNA Learning Center, all the great research that we have going on all across our several campuses. So thank you very much, Amanda. This is awesome.
It was a pleasure. Thanks for having me.
It was a great pleasure. And thank you, everybody else. Good night.